So now that we know how to look at monohybrid crosses just with one characteristic and do things like test crosses, we need to look at dihybrid crosses. So dihybrid means two, so di means two and hybrid means cross. And again, we'll have a classic P generation, F1 generation, and F2, but now we have additional characteristics. So we have smooth or wrinkled, and yellow and green. And in this example, we're crossing smooth yellow P's with wrinkled green, and that's our P generation, so those came from true breeding plants. When we cross those, we get all smooth and yellow. So that tells us that smooth and yellow are dominant because they show up in the F1 generation. So we know that smooth and yellow are the dominant characteristics. If we allow those to cross, so two of these smooth yellows from the F1 generation, our F2 generation, we get the ratio of 9 to 3 to 3 to 1. 9 of them will be the smooth and yellow, so the two dominant traits. 3 of them will have a dominant trait and a recessive trait. In this case, they're yellow, and that's the dominant trait, but they're wrinkled, a recessive trait. The next 3, it's switched, so now these are green. That's the recessive trait, but they're smooth as the dominant trait. And 1 will have both recessive traits. And again, we can walk through this using Punnett squares, and your book uses capital S, lowercase s for smooth and wrinkled, capital Y and lowercase y for yellow and green. It's really hard to tell those apart when I'm writing them because the s's kind of look the same and so do the y's. So in my example, I'm going to use just A and B. So smooth is going to be capital A, wrinkled is little a. So capital A, little a. Yellow is going to be capital B, green, lowercase b. And when you guys are doing these in class or as homework or as part of the exams, if you change the letter that you're using, or if I don't give you a letter and you need to add one, just make sure that you're using the correct terminology if it's dominant and recessive. And the specific letter is not important, but you always have to give me a key, so tell me what they mean. So we want to do this cross, and we want to start with our P generation. We had smooth, true breeding, and yellow. So this is smooth and yellow. And we cross that with wrinkled, green seeds. If we do a Punnett square, we need a slightly larger square now because we're doing dihybrids. So I will draw a slightly larger square. And again, one parent's going to go down the side and one parent across the top. For the first parent, this A is going to separate from the other A, and it might go with the first B. So we could have A, B. Or this A could go with the second B. We're looking at possibilities here. This A could go with the first B, or that A could go with the second B. You can see we have 4 possible ways we could combine the A's and B's, so there are 4 slots on its side, even though they all look the same. For this parent, you'd have the same thing, so first A, first B. Outside A, outside B could go together. Inside A, inside B could go together. Or last A and last B could go together. You can see that they're still all the same. And then you pull the letters in. So big A comes across, little a comes down, big B comes across, and little b comes down. And all of the squares here would be the same because it's all the same for the parent. So it would be capital and lowercase a, capital and lowercase b. So our F1 generation is all heterozygous. And we can cross two of our F1s together. So if we draw a second Punnett square to represent our F1s crossing, again, one parent would be A, B, B, and the other parent would be A, B, B. One thing I want to mention here, if you've ever used the FOIL method in like a math class, I know that's fairly common. So FOIL is first, outside, inside, last. And that can help you keep track of which A's and B's you've crossed. So we'll put this one down the side, and we'll put this one across the top in this Punnett square. So our first A could go with the first B. So my first possibility, let me get my square set up. My first possibility for this parent would be a capital A and capital B. Second possibility would be the outside A and the outside B. So it would be a capital A, little b. Third possibility is inside A, inside B. So in that case it's a little a and a capital B. Or last A and last B. So little a, little b. We'll do the same with this parent up here. So first A and first B. So A, B. Outside A and outside B. So A, B. Inside A and inside B. A, B. And last A and last B. And that gives us all the possibilities. This represents what's in the sperm and the egg and the parents. And then we pull them into the middle to represent the offspring. So we'll have big A, big B, big B. I recommend that you organize it, so if there are capital and lowercase letters, you group the A's together and you group the B's together and list them capital first, because when you do crosses, really what you want to know is the end result. So you'll have to come back through here and find the phenotypes of all of these. It's easier to see that this one's got capital A for smooth and capital B for yellow if it's organized. So try to organize them as you transfer them down. So capital A from this parent, little a from the parent coming down, two big B's. And then you can continue filling in the Punnett square for the rest of them. This is the example that your book gives. So we have capital S's and Y's again, lowercase s's and Y's. That F1 generation, capital lower, so heterozygous. And this shows the F2 generation, so the four possibilities for the egg and the pollen or the sperm. And when you pull them all into the middle, you can see 9 of them. So let me find all 9 of them. So these 9 are all smooth and yellow. Then you have 3 that are wrinkled, or excuse me, green and smooth. So there's 3. And then there's 3 over here that are wrinkled and yellow. And then there's just 1 that's wrinkled and green. So that 9, 3, 3, 1 ratio. The main part of this is being able to get the parents correctly listed on the outside with their 4 possibilities. With dihybrid crosses, it shows us Mendel's second law. Mendel's second law is the law of independent assortment. So for our traits, smooth and yellow and wrinkled and green, they all move independently. So smooth and yellow are not connected, wrinkled and green are not connected. They're like separate Legos. So maybe we have our yellow and our green characteristics, and maybe that's smooth and that's wrinkled. But when they're passed to the offspring, they can all separate and assemble in those different combinations, which gives us our 9, 3, 3, 1 ratio. Another example, this is one that your textbook gives. It shows dimpled chins and albino individuals, and they both assort independently. So having a dimpled chin is not connected to being albino. So you again end up with a dihybrid ratio. There is an exception to dihybrid ratios, and that comes with linked genes. So all of these genes are on chromosomes, and they separate during cell division, and crossover helps mix the genes on individual chromosomes. However, if you have two genes located very close together, it's unlikely that crossover is going to separate them because they're so close together. You can think of like Legos stacked, and you just can't get them apart because they're so tightly packed together. So some genes are linked. An example of that would be red hair and freckles. They tend to be linked and go together. It's hard to separate them genetically because physically on the chromosome, those characteristics or traits are close together. So that'd be an exception to Mendel's second law. Some other exceptions to things that Mendel found are things like incomplete dominance. So incomplete dominance, you don't have one dominant one that completely hides the other. Your heterozygous individual is an intermediate, somewhere in between the other two. This is common in lots of flowers, including snapdragons. And because it's not completely dominant and completely recessive, you can't just use capital and lowercase letters. Because remember, when we first introduced the capital and lowercase letters, by definition, the capital letter means that it's completely dominant. And the lowercase letter means, by definition, it's completely recessive. So you can't just use capital and lowercase letters because that doesn't show the incomplete dominance. Instead, you can use a second letter to indicate that they're at the same location on the chromosome, so they're alleles, and then use a superscript for your specific color. In this case, we're using C, and if we draw a pair of chromosomes up here, the C tells us that they're at the same location. So this is location C. If I just use two different letters, if I just use a W and an R, that means that they have to be at two different spots on the chromosome, and you need to do a big dihybrid square. But because that C puts them at the same location, it's just a monohybrid cross, so simple squares. If we have a P generation, true breeding white, the color is white for both genes, crossed with the color is red for both genes. If I put that into a simple Punnett square, because that C tells me it's at the same location, I have white genes and color red genes, so all of the offspring will get a white and a red from their parents. And because the heterozygote is intermediate, if you mix red and white, you get pink. So all of the offspring of these parents would be pink. Now if we continue, these offspring in the middle here are the F1, so pull those out and cross a couple of F1s to get our F2 generation again, a second Punnett square. We'll put this parent down the side, so it'll have a color is white gene and a color is red gene. This parent across the top will give a color white gene and a color red gene. Pull them to the middle. This one will get two color white genes. This one will get a color white and a color red. This will get a color white and a color red. And this one will get two color red genes. So now we have a 1 white to 2 pink to 1 red ratio. So a 1 to 2 to 1 ratio, because we can see these heterozygous individuals in here. And that's incomplete dominance. And here's the figure from your textbook that shows crossing our white and our red gives us pink. Taking a couple of these pink ones out and crossing them gives us 1 white, 2 pinks, and 1 red. So 1 to 2 to 1 ratio. You can also have codominance, and in codominance, both traits are expressed. They're not mixed. And this is a cute picture, and we'll look at some real examples. So we have parents. Dad's got the horizontal striped shirt, Mom's got the vertical striped shirt. The offspring is neither horizontal stripes or vertical, but a mixture of the two, so both. And that's a cute representation of co-dominance, where both characteristics show up equally. An example of codominance shows up in blood type, and it also has multiple alleles in blood type. So with blood type, we have type A, type B, type AB, and type O. The differences in those blood types really is just sugars or antigens hanging on the outside of your red blood cells. So type A has a specific shape, in this case little balls. Type B has a different shape, little triangles or pointy ends. If you're type AB blood, you have both the little round shape and the pointy shape. So that's a mixture of both. It's not an intermediate. They both show up. They're co-dominant. If you have type O, you actually have neither of those particular sugars on your red blood cells. To represent these, we have to use a similar method to what we did with our incomplete dominance and use superscripts. So type O is recessive, so it can have little letter I. Type A, you use I as the base letter and put the blood type as the superscript. So A could either be homozygous for A, so I capital A, or it could be heterozygous and hiding blood type O, so I capital A, little I. B blood types could be the same, IB, or IB hiding little I or blood type O. And for type AB, you can only have an IA and an IB. And those were the different abbreviations for the blood types. Make sure that you know how to use the superscripts for these, because on the exam, if you just write A's and B's, I'll take off points. You need to have them as superscripts to indicate that they are codominant and not just dominant. Also, because of these different sugars on the outside, that's why you have to get the correct type of blood when it's donated, because if you get the wrong type of blood, antibodies in the person will attack it. So you can look through this and see which type of blood you would be able to give or receive. So let's look at a question practicing blood types. And we'll look at a real-life case. Charlie Chaplin, if you've heard of him, he used to be very common in silent films back when TV was a brand new thing in movies. But Charlie Chaplin, in 1941, he had been dating a gal, and they had been separated for about 21 months, and she had a baby girl. And the mother came back and sued Charlie for child support. Said, well, Charlie Chaplin's the dad of my daughter. And they went to court, and Charlie challenged it. They went to court, and at that time, blood typing was a brand new thing. And they found that Charlie Chaplin had blood type O. So how could you represent blood type O? If you remember from the previous slide, that would be 2 little i's, correct? And Mom had blood type A. And there are 2 ways you could be blood type A, right? You could have I with capital A, I with capital A. Mom could be that. Or Mom could be A and hiding the little O. So those are the possibilities for Mom. And the child had blood type B. So I'll let you look at what letters the child might have. What are the possibilities for the child? It'll be very similar to what Mom is, just replace the A's with B's. based on this, do a Punnett square. Actually, you'll have to do two. You'll have to cross Charlie and the first possibility for Mom, and do a second Punnett square and cross Charlie again with Mom's second possible blood type. Should Chaplin pay child support to this little girl? Never mind the fact that they've broken up 21 months prior to that, but should he have paid child support? So on a sheet of paper, go ahead and try working through two Punnett squares, one for each of Mom's possible blood types, crossed with Charlie up here, to prove your answer, either yes or no, and then prove it using Punnett squares. And keep this in mind when you come to class on Tuesday.